Recording. Okay, quick recap, right? So we have finished talking about the neurons. We have talked about axonal transport, right? We have talked about the different types of synapses, dendrites, axons. Could you name me the three functional types of neurons, please?
Motor, intermediate, sensory.
Motor, interneuron, and sensory. And what are the morphological types of neurons?
Bipolar, multipolar, unipolar.
And pseudo-unipolar, right? So motor neuron is which kind?
Multi.
Perfect. Inter is also multi, right? And sensory is
Uni.
pseudo-uni. Okay, so I apologize, I should have changed it there, but it's actually pseudo-unipolar. Yeah. Nissl bodies, what can you tell me about them?
Ribosomes and the AR.
Nissl body.
Yes. So Nissl bodies are packed ribosomes, packed RER. And why is nucleolus visible?
It has a lot of nissls, a lot of rough endoplasmic reticulum, and ribosomes.
So nucleolus is visible because that's the place for the ribosomes production, right? So that's where we're going to be making it. But yeah, correct. Okay, so we have talked about neurotransmitters, right? So you remember I told you to look at them. So what do they do? What are their functions and etc. Axonal transport, we have kind of mentioned it before, right? So don't forget, inside of the axon, we have microtubules, and you can see that there are lots of things which are going in different directions. How do we call the movement when it is from the nucleus to the axonal terminal? How do we call it?
Anterograde.
Perfect. We're going to call it anterograde. So this is an anterograde movement, and backwards is the retrograde movement. Pretty much repetition. The idea is the same, that we have the motor proteins, right, that are moving in different positions. So you have to remember that anterograde is kinesin and dynein is the retrograde movement, right? So it's the repetition of the histo 1, right? So the different charges that are driving them and etc. Could you tell me what is the charge of dynein?
Positive.
Perfect. It has to be positive because it is going in the retrograde movement, right? So it's going backwards, it's going to the nucleus. Nucleus has a negative charge. So of course, dynein has to have positive charge to kind of go towards the nucleus. The kinesin will be the opposite, right? So to have a negative charge because it is going away from the nucleus, it is repelled by the nucleus itself. I guess this is important because we have two types of transports. We have the slow transport system and the fast. Okay, so the difference between them, it's understandable. One is slow, one is fast. Just what exactly is being transported by the fast system? So a fast anterograde will be transporting, you're going to see, so some kind of sugars, amino acids, calcium, right? So mitochondria can be transported fastly, different organelles, and etc. Neurotransmitter as well. I don't think, well, synaptic vesicles, they are filled with the neurotransmitters. So neurotransmitters, the way they are transported from the cell body all the way to the terminal end, it is the fast anterograde. Everything else hypothetically could be transported back as well, but that typically is not the case because neurons tend to use up everything that is being transported. So rarely some things will return to the cell body, rarely. Majority of the time, everything is being used up. Okay, let's talk about Schwann cells a bit. So you remember that Schwann cell is a supporting cell in the peripheral nervous system. It is responsible for myelination of the axons in the PNS, right? So you can see that this is a neuron in the PNS. Its axon is fully kind of surrounded by the Schwann cells. Don't forget that Schwann cell is a faithful cell because, one Schwann cell per one axon, right? You can see that it just stays on one axon. So in the peripheral nervous system, you can find a bunch of Schwann cells on one axon. We have talked about that Schwann cells are arising from the neural crest cells, but don't forget from the last semester that lots of things come from the neural crest cells. So how do we know that Schwann cell is the one which will come from there, right? So it has to be SOX10. SOX10 is a transcription factor. So these neural crest cells that express SOX10, they are destined to become Schwann cells. So for the exam purposes, SOX10 neural crest cell equals Schwann cell. So to be a Schwann cell, you need SOX10. Let's talk about how do the Schwann cells make myelin sheath. So this is a very good example here. It's kind of showing you an overview of myelination, right? So this is the axon itself. On the surface of it, you can find the Schwann cell. And you can see how Schwann cell is sort of wrapping itself around the axon several times, creating myelin sheath. So in a way, myelin sheath is a plasma membrane of the Schwann cell, right? It is a entire Schwann cell itself that is just sort of wrapping around the axon. There are different domains that we need to talk about. So that's for the next picture. You can see that this is the axon itself, and this is the Schwann cell. At first, you see the Schwann cell starts to wrap itself around the axon, and as a result, we can see three domains. So the outer part of the Schwann cell, which is not facing the axon, this is called ab-axonal domain. That part of the Schwann cell which is facing the axon, this is known as the ad-axonal domain. And the part where two meet up, this is known as the mes-axonal domain. So which of the domain is responsible for creating myelin sheath? You can see on picture C. Which domain is the one which is wrapping around?
The ad.
It's going to be the mes-axonal domain, which is wrapping around, okay? You can see this is the axon. This is the mes-axonal domain, and it is the one which starts to coil several times. It's like if I have to show it with my hands, it's very difficult, but you do this, okay? So you see that only one mes-axonal domain starts to like coil several times until it creates this entire myelin sheath. So I know that typically we say myelin sheath is made by the Schwann cell, but in neuro, you have to be a bit more specific by which part of the Schwann cell exactly is it created. It is created by the mes-axonal domain, by this junction, and not by the two, because do you see this is the inner, this is the outer. So only one mes-axonal domain is responsible for coming in and wrapping several times. The other one stays outside. So again,
I have a question, sir. The mesaxon is just before the wrapping, right? During the myelination, it disappears after that.
Exactly. So it stays as it is. So exactly. So it's going to be, it does disappear because all of these rings will fuse together. Yeah. So these mesaxonal rings, they'll fuse together and create that membrane. Yeah. So if we show with our hands, right, this is mesaxon, this is also mesaxon, this is my Schwann cell like that. So only one mesaxonal domain will start to wrap several times, creating the myelin sheath itself. What regulates the thickness of the myelin sheath, right? So how do we know how thick should it be? A certain protein, which is called neuroregulin, okay? And neuroregulin is made by the nervous system itself. So it's not the Schwann cell that regulates the thickness. It's the nervous system itself. So the neuron itself will regulate its myelination extent. Neuron itself will secrete the neuroregulin and regulate the thickness of the myelin sheath. How exactly? The bigger diameter of the axon, the thicker the myelin sheath should become. So the bigger the axon, the more myelin sheath, the more neuregulin in the first place, right? Okay, so what can we find within the myelin sheath? So this is more or less like a composition, right? So of the myelin sheath, most importantly is that you see it is a plasma membrane of the Schwann cell. So phospholipid bilayer times several, right? You can see that because it is ROPin several times, it's a phospholipid bilayer. But you can find some kind of proteins there. Okay, so we'll talk about them right now in a sec. There are a couple of proteins there as well. Okay, so node of Ranier we have talked about, mesaxon we have talked about, tra-ta-ta, trulala. And yeah, okay, so a question. What is wrong with this picture?
The compact myelin, the basal lamina.
Oh, actually, you're right. This should not be there. It has another name. We'll talk about it.
External lamina.
Very good. So this should be external lamina. This is not a basal, this is external lamina because only epithelial cells possess basal lamina. Schwann cell is a non-epithelial cell, so it does possess external lamina on their surface. But one more. A certain domain is actually absent, the mesaxonal domain. Okay, you cannot see it here. So again, you should be aware that at first you will see two mesaxonal domains, and only when one of them starts to wrap around, only then this myelin will be created. Okay, so only then. Okay, whenever we say unmyelinated neurons, we always mean that there is no myelin sheath, right? So they don't possess the myelin sheath. But the question is, do they have the Schwann cells?
Yes.
Yes. Okay, so that's the answer. So absolutely, they will possess the Schwann cells. You will find them there, but you can see that the position of the Schwann cell is very different. So you can see that maybe this particular Schwann cell tried to be oligodendrocyte. It tried to myelinate as many axons as possible. So you can see that it is too much. So one of the mesaxonal domain is not capable of going around, right? So it's too much. So it can't possibly wrap around it. Or the same idea is here, just from two different sides. So most important things is this, that unmyelinated axons, they still have Schwann cells. They still are covered by the Schwann cells, just not entirely. The myelin sheath is absent, right? The myelin sheath, 80% of it is lipids. A very stupid question, why? Why majority of myelin sheath is lipid?
Lipids
Because
are
during the
insulative.
leakage of the action, like of the signals.
But like structure-wise, why 80% of it is lipid?
Because of the phospholipids that are
Exactly, yes.
Me
Okay,
too.
because don't forget that the myelin sheath is the Schwann cell plasma membrane wrapped several times. So of course, it's just a bunch of phospholipids, cholesterol just being stuck together. So more than 80% of it will actually be lipids. Okay, node of Ranier, you remember, right? So that's the gap. So you remember this. Okay, so let's take a look at the histological representation of the myelin sheath. So what you can see here are a bunch of axons. The white space that says A is the axon. But in our university, they don't say axon, they say axoplasm. So in the exam, you will see axoplasm, the cytoplasm of an axon, right? The bluish tissue that you see here, that's the myelin sheath, right? So right on the surface, you can see that there are a bunch of myelin sheath present there. Would you be able to see myelin sheath with H and E? Yes, no, why?
No,
Why not?
Because it's majority lipids and lipids being washed
Very
out.
good. Very good. So don't forget H&E preparation. Lipids are not going to be visible whatsoever. So if you were to see myelin sheath, what would you use?
Posh.
Heavy metals.
Heavy metal. PAS is for glycogen. It's for glucose-rich molecules, right? So if you wanted to actually see the lipids of the myelin, you will definitely have to use heavy metals. Two heavy metals that you could possibly use: silver or osmium. In our university, our slide is osmium. Okay, so we're actually going to be using osmium to see the lipids. Satellite cells, these are the supporting cells in the PNS as well, but they do not surround the axon, they surround the cell body. They are typically small cuboidal cells, nothing interesting about them, right? So just outside of the cell body of a neuron, you can only see the nuclei. Because they're supporting cells, just blah, blah, blah. So metabolic exchange, right? So electrical insulation of some sort, and etc. So very similar to Schwann cell, except for the myelination. Cell body is not myelinated, right? Because there aren't any Schwann cells there. Okay, so here, okay, I will return to this. There is a better way to describe it, but okay. Well, actually, no. Which cell is analogous to the satellite cell in the central nervous system? It's going to be the astrocyte. Okay, so if you were to ask yourself, like, what is the satellite cell in the CNS? That's astrocyte. What is the Schwann cell in the CNS? That's oligodendrocyte. So just try to compare the two cells with the two nervous systems, right? So remember that satellite cells, they are basically astrocytes in the CNS itself. Where would the satellite cells come from? The same as the Schwann cells, they'll come from the neural crest cells. Maybe you remember from the last semester, neural crest cell gives to the entire PNS, the entire peripheral nervous system with whatever it has there, ganglions, nerves, supporting cells, all of those things will come from neural crest cells. Different transcription factors, of course, involved, but it's not important. Remember the SOC stand for the Schwann cell, and you will be fine, because that's what they usually ask. Okay, there's something that they asked last year. It was this. Another name for this is a myelin. I don't know how to pronounce it. In seizure. I don't know. Incisure. Okay, so how do you pronounce this? Myelin, what? Incise, okay, this. Okay, so incisure. Okay, so that's another name for Schmidt-Lantermann cleft. So what they have done on this picture, you can see that they have unwrapped the Schwann cell, right? So this is the axon, and they have fully stretched it out. This picture is trying to tell you that the myelin sheath is not uniformly distributed. There are some gaps where you have less of the lipids than in the rest of the myelin sheath. Yeah. And those areas, those defects in the myelin sheath, they have a name. They're called myelin incisures or Schmidt-Lantermann cleft. So these are areas that possess less lipids than the rest of the myelin sheath. On this picture, they're drawn very nicely. Like you see exactly straight line here, straight line here, straight line here. In reality, that's not the case. In reality, these areas are patchy. There's a bit of it here, a bit of it there, and etc. I repeat, it's just a defect in the myelin sheath itself. Because think about it, as this myelin sheath is wrapping around the axon, there is unequal pressure everywhere. It's like you're squeezing the lipid a bit there, a bit there. So that creates the gaps where lipids are not present. Okay. Don't get me wrong, they are present, just not like 80% amount. Okay, so let's take a look at the structure. First of all, what's the staining?
It's azan.
Perfect. This is going to be azan staining. What are these?
cell body.
Yes, so these are going to be the cell bodies of a neuron. So how do you call this entire thing?
Axon.
Sorry, nucleus ganglia.
Yes, either nucleus or either a ganglion, but this is in the peripheral nervous system. So this is actually a ganglion. So you can see a bunch of cell bodies, right? And together, so this is a ganglion. You magnify a little bit. How do you call these cells on the top of the neuron cell body?
Satellite.
Satellite cells. Yeah. Okay, so these are the satellite cells. Let's move on to the CNS. We talked majority about them last class. So you remember that oligodendrocytes, astrocytes, microglia, ependymal cells. So the same idea that as the last class, so myelin sheath formation, supporting cell, macrophage, CSF production, right? So we're producing the cerebrospinal fluid. The same idea as with the PNS, you cannot see the entire cell. Typically, you can only see a nucleus. It's very difficult to find the entire cell. You will need heavy metal, so silver or osmium, and etc. This is one question in the exam. The question is, what comes first, neurons or neuroglia? You remember this picture? So
Yes.
neural tube, right? Neural crest, paraxial, right? Notochord, two dorsal aortas are here. So a question, what comes first? Neurons or neuroglial cells?
Neuroglial.
Perfect, yes. So neural tube, you remember the process is called neurulation when you're creating the neural tube. During neurulation, neuroglial cells are produced first. These neuroglial cells are going to be creating some kind of scaffolding, okay, like a scaffold, something like a barrier. And because of this, the neural tube is exactly as it is. So think about it this way. This future neural tube first makes the supporting cells. Supporting cells create a barrier outside. And thanks to this barrier, your neuroectoderm is folded into a neural tube, as we know. And the neural tube in you, that's your brain and spinal cord. So yes, neuroglial cells are appearing before the neurons, before the entire CNS. Okay, astrocytes. These are the most common, the most dominant neuroglial cells in the CNS. There are two types of astrocytes. We have the protoplasmic and fibrous astrocytes. The difference between them is the location. So protoplasmic astrocytes are sitting in the gray matter. Fibrous are sitting in the white matter. Protoplasmic astrocytes have a very short branching cytoplasmic processes. So you will see it. Fibrous has relatively straight. So they're not branching. In the brain, which one is the outer, gray or white?
Gray.
Gray, okay, gray is the outer, white is the inner. So you should know that the protoplasmic are going to be the outer and the fibers are going to be the inner. In the spinal cord, it's the opposite, right? So you're going to see that it's going to be the opposite. White is the outer, gray is the inner. Why is the fibrous astrocyte called fibrous? Because it contains this intermediate filament, which is called glial fibrillary acidic protein, GFAP. It is an intermediate filament type 3. I don't know if you remember, in Histo 1, we have learned that there are 5 types of intermediate, 5 classes of intermediate filaments. Class 1 and 2 are keratins. Class 3 is this one, GFAP. So this GFAP is used to identify fibrous astrocytes. I think we even have a slide, like later when we do the brain, we even have a slide where we used antibodies against the GFAP to show the fibrous astrocyte specifically. So this is what you can see. So this is an astrocyte. Guess which one? Is it protoplasmic or fibrous?
Fibrous.
As you can see, it's
Protoplasmic.
yes, it is protoplasmic because it is branching. You see, there are lots and lots of different like tree-like structures. So it is definitely branching. So this is a protoplasmic astrocyte, which is present again in the gray matter, in the most outer. Notice that it is always between a neuron and a blood vessel. Because you remember from last class, what is the most important job of the astrocytes?
Blood-brain barrier.
Perfect, BBB, blood-brain barrier. So you're going to see that always they're going to be right next to the blood vessel. Yeah?
Can I see that the feet also?
Not like that. So this is a very good picture because it was it is immunohistochemistry. So we use exactly the antibodies, fluorescence, okay, to highlight the entire thing. In our slides, not really. You only see the nucleus. Okay, so okay, there is one staining that you can see, but majority of the time, like you won't be able to see this. There's only one slide where you can see it, but for the rest, you will only see this, just the cell body. That's it. Okay, this one, what do you think?
Cell body with dendrites and axon.
Nope. We're talking about astrocytes. There are two types of astrocytes. This one is the protoplasmic because it is branching, right? But this one has a straight ones. So this is a fibrous. Yeah, this is a fibrous astrocyte. You will see these a lot. Okay, so these ones you will see a lot. Again, with specific staining. So not H&E, yeah? So only with heavy metals you'll be able to see the processes like that. Okay, what does it contain? Fibers, what does it contain?
The acidic.
Yes, okay, so this acidic thingy, the GFAP, glial fibrillary acidic protein, which is a intermediate filament class 3 again. Okay, so we have said that astrocytes, BBB, right? So they play a super important role in movement of metabolites, right? So you can see that their food processes are always on the top of the blood vessel. How exactly do astrocytes create the blood-brain barrier? The food processes of the astrocytes, as you can see, are landing on the wall of a blood vessel. This induces formation of a tight junction. So the only reason for tight junction to be here is because it is induced by the food process of the astrocyte.
When tight junctions between what?
Between endothelial cells. There are two endothelial cells of the blood vessel. Yeah. So it's like this food process is trying to seal the blood vessel, right? So it's trying to close it to make sure that the wall is not leaky. So it creates a tight junction. Could you tell me how exactly it induces formation of tight junction? Like which proteins are forced to appear here between the two endothelial cells?
Claudins.
Perfect. Claudin.
Claudins and claudins.
And JAM. Okay, so don't forget this is a blood vessel. So JAM will also be there. Occludin, claudin, and JAM, junctional adhesion molecule, right? So those are the three proteins that the food processes of the astrocytes are inducing to appear. So that's the role of the astrocytes in the BBB. In addition, these food processes are very special because they are not only trying to kind of seal up the holes in the blood vessel, they're also trying to seal up the holes on a neuron in the CNS. So hypothetically, this food process could close or seal the node of Ranvier. It can fully close it like that to make sure that action potential continues to propagate. So this astrocyte, I repeat, they're the most numerous glial cells in the CNS, and they're also the most important because they're making sure that all of the ions, sorry, I thought like that, all of the ions are going in the normal direction, right? So they're making sure that action potential propagates, and they're making sure that nothing is leaking out of the blood vessel.
I have a question, sorry.
Mm.
You said that it seals both the endothelial cells and the cells of the neuron cell, but how does it allow the movement of metabolites then?
Very good. So you will see it. Okay. So when we talk about the BBB, you will see. So it is going to be part of it. So only little things can diffuse. So only oxygen can diffuse. Only carbon dioxide can diffuse. Okay. So big things like proteins, hormones, they can't. They won't be able because the food process is right there. Yeah. So only little tiny things can actually come out.
Can glucose pass?
Of course, glucose, biochemistry 101, there is a transporter. Okay, so the GLUT1 transporter. So technically, you know, it will do it on its own. The same for amino acids. They have transporters there. But simply to diffuse, right? So not like being transported, simply to diffuse. It's just little gases. That's it. Just gas can diffuse. The rest of the things cannot. Yeah. Okay, so take a look. This is a piece of a brain, right? So we're looking at the most outer part. So the most outer is definitely the gray matter. So within the gray matter, you will find the protoplasmic astrocytes. Protoplasmic astrocytes of the gray matter of the brain are very special because you can see that they're sending their food processes all the way to the pia matter. You remember, you probably know this from the general knowledge, right? The three matters of the brain, dura, arachnoid, and pia, right? They're like meninges that are covering the brain. So the protoplasmic astrocytes, in addition to sending their food process on a blood vessel, in addition to sending their food process on a neuron, it sends another food process on a pia matter. And a bunch of these food processes together that are sort of attaching to the pia matter creates a membrane, which is called glia limitans. So glia limitans is additional layer under the pia matter, and it is created by the food processes of the protoplasmic astrocytes specifically. Why not fibers? Fibers are deeper. They're in the white matter. They're much deeper. So it has to be protoplasmic astrocytes. Can you see this layer in anatomy practical? No. Okay, it's a very tiny thing. You will not be able to see this. This is more of a theoretical layer, but it is important to mention it. Okay, so glia limitans is a very important layer. So if we do all the layers, scalp, right? So the skin that we have here, cranium, skull, dura, arachnoid, subarachnoid space where we have a bunch of blood vessels, pia matter, and then under it, glia limitans. So quickly, who creates it?
The proto.
Fibrous astrocyte.
Not fibers, because fibers are deeper. Fibers are in the white matter. White matter of the brain is deep. It's the food processes of the protoplasmic astrocytes
Doctor.
because of the gray. Yes. Okay.
I have a question. Is this one that will give the internal and external?
Okay, hold on. Yeah, there's something else. You're going a bit way ahead. Okay, so yeah, sort of. Okay, so we'll talk about it right now. So what is the job of the glia limitans? What do you think? What does it do?
Divide into two?
No, it doesn't divide into two. I know that you heard external, internal, but it's different things. Okay, so they're different things. It doesn't divide. What is the job of the glia limitans? Okay.
It's SNF, CSF.
CSF is produced by the ependymal cells. These are the protoplasmic astrocytes, so not the same. Glia limitans, the word itself should be like important for you. It's limiting something, right? So it creates a limit of some sort. Glia limitans prevents migration of neurons into the meninges. It makes sure that the neurons do not send their axons through the meninges someplace else. Okay, so it creates some sort of barrier that makes sure that there is no over migration, because guys, honestly, neurons are very, very fast cells. Okay, so they synapse like this. They find a way to synapse with anything. So that's why we need something that keeps them in their place. So that's where the glia limitans comes in. It prevents over migration of neurons to make sure that they stay in the brain. They don't send their axons to the meninges or like outside of the meninges somewhere else, you know? So yeah. Okay, oligodendrocyte, we have talked about it. So this is a Schwann-like cell in the CNS, right? Myelination. Don't forget it is a very promiscuous cell. So you're going to see that it sends a bunch of food processes to all the different axons. Do you think it's going to do a good job of myelinating? As you can
No.
see, it's busy with so many axons. So as a result, it doesn't do a good job. I mean, it does an okay job. Majority of axons are myelinated, but some of them aren't. So now we reach the most important thing. We have to compare the Schwann cell and the oligodendrocyte. So what are the main differences between them? Of course, CNS, PNS, that's understandable. And this is the thing that they will ask you in the exam. We said that myelin sheath is 80% lipids. The rest of the percentage is going to be some proteins. And one of these proteins are called P0 and PMP22. P0 and PMP22 are limited for PNS. So only in the myelin sheath of the PNS, you can find these proteins. In the CNS, so produced by the oligodendrocyte, myelin sheath there, you will not find P0 or PMP22. Instead, you will find a bunch of proteins and glycoproteins. You can see them here, right? So proteolipid protein, myelin oligodendrocyte, or oligodendrocyte myelin glycoprotein. Like, I'm not sure who was naming things, but great job. Okay, so these are two different proteins. Just switch the two names, the two words, and completely two different proteins. So these things are limited to the myelin sheath of the CNS. Schwann cell possessed external lamina. Remember we saw it previously, right? So there was additional layer on the top of the Schwann cell. It was external lamina. External lamina is like a basal lamina, right? It is like a basement membrane of the non-epithelial cells. Oligodendrocytes do not possess external lamina. So if we try to imagine it, right? So this is my axon, and this is my myelin sheath, and this is my other axon, and this is my other myelin sheath here. If they're covered, if these two axons on the top are covered by the external lamina, would they stick to one another or no?
No.
They will not stick to one another, right? Because the lipids are not visible, like they're hidden, they're under the external lamina. But if you do not possess external lamina, what will happen to the two things?
They all stick together.
They will stick together. So axons in the CNS are always sticking to one another because they do not possess the external lamina. The two lipids, you're going to see, the two myelin sheaths of two different axons are attracted to one another. They stick to one another. Okay, so it's going to be very, very sticky. Nodes of Ranvier in CNS are much larger, so that's why the saltatory conduction is much more faster, much more bigger, etc. Unmyelinated neurons in CNS are bare. What does it mean? It means that they do not possess oligodendrocytes on their surface. So remember we talked about it. In the PNS, unmyelinated still had the Schwann cell on their surface. It was unmyelinated, but it still had the Schwann cell. In the CNS, oligodendrocyte is busy myelinating many axons. So if there is no myelin, how can one oligodendrocyte be on the top of several axons. It cannot. So that's why you will see that unmyelinated axons in the CNS are bare, no oligodendrocyte present. Okay. I should ask you a question here. Yes. Could you tell me why action potential travels faster in the axons with a greater diameter?
Because they have a greater myelin sheath and better insulation.
Good way to think. Okay, so it was my fault. Without using the myelin sheath, without talking about the myelin sheath and
Because
etc.
less resistance when you have bigger area, less resistance.
Who creates the resistance?
I know that there is a link between the radius and the resistance. The bigger the radius, the less the resistance.
I know, but who creates the resistance? You're absolutely right. The bigger the diameter, the less the resistance. My question is who creates that resistance?
Maybe the channels, ion channels.
Not channels?
The
Ion
basement membrane.
extracellular ions in this.
Intracellular things. Okay, so you guys are close. I will explain. So this is the axon. What is inside of an axon?
Microtubules.
Microtubules. And these microtubules, on the top of them, were dynein and kinesin. So when the ions are traveling inside, what is the probability that they will hit these structures if you have a smaller diameter?
High.
High, super high. Let's imagine I have an axon with a greater diameter. The same idea, microtubule, dynein, kinesin. What is the probability that ions will hit things?
Lower.
Super low.
Super low, even better. Yes. Okay, so super low. So the resistance that you're talking about is created by the things inside of the axon. It could be anything: microtubule, neurofilaments, organelles, because maybe dynein or kinesin is carrying mitochondria here, right? So just whatever is present inside of the axon, it can create a resistance. Okay, microglia are phagocytotic cells. Nothing interesting. We almost never see them because just 5% of them are present there. They have a super short twisted processes. Processes have spikes, whatever. We have said that there are 4 neuroglial cells in the CNS: oligodendrocyte, microglial cell, astrocyte, and ependymal cell, right? So 4 of them. Do you think microglial cell will come from the same thing as the rest of them, or not?
No, no.
Why not?
I really don't know.
Anybody?
Probably from the blood.
Perfect. Microglial cell has to come from the blood. Why?
Because they're phagocytotic and those cells tend to come from the blood.
But because we're second year, we're going to say not blood, we're going to say bone marrow precursor, because remember, all of the blood comes from bone marrow. So you're going to see that microglial cell, because it is a phagocytotic cell, it does not have the same origin as the rest of the neuroglial cell. Instead, it is going to arise from the bone marrow monocyte precursor, because phagocytes come from monocytes. Fun fact, if you have some kind of injury or disease, doesn't matter PNS or CNS, the microglial cell becomes super active and it starts to digest the myelin. It's like imagine I cut myself and I cut the neuron, yeah? There is a damage in myelin that activates my phagocytotic cell and it starts to create more damage by phagocytosing the rest of the myelin. So that's why whenever you have neurosurgery, Like if there was some kind of injury, the surgery should be within hours, because you have to make sure that the phagocytotic cells are not doing their thing. You have to like fully suppress the immune system and quickly repair things, because phagocytotic cells become super activated when there's injury or disease or trauma. So they do more damage than the injury or disease itself. Ependymal cells, CSF, okay, nothing interesting. Choroid plexus, you remember, right? So ependymal cells plus the blood vessels, because CSF will be arising from the plasma of a blood vessel. There is a particular specialized type of the ependymal cell. It's called tanycyte. Tanycyte have a very particular location. They're present in the floor of the third ventricle. I'll explain it in a sec. They don't possess cilia. Okay, so these cells do not possess cilia, and they're responsible for kind of transporting things from the CSF to the blood. So there are some things that may be too much, maybe glucose. Okay, so that's the area where you will pump the glucose from the CSF to the blood. Where is the third ventricle? So you can see that this is your CNS, and these things are the ventricles which are filled with the CSF. We have two lateral ventricles. Two lateral ventricles have two horns, right? So we have technically three horns, the anterior, posterior, and the inferior horn. So that's one ventricle. We have the third ventricle, right? We're going to have the fourth ventricle, and we're going to have the central canal of the spinal cord. So these areas are filled with the CSF. Where is choroid plexus? In the lateral ventricle. So roof of the anterior horn of the lateral ventricle, that's where choroid plexus will be. That's the area where we produce the CSF. Third ventricle, which structure is nearby?
Pineal gland.
Pineal gland will be here. Pituitary will be here.
Thalamus.
Thalamus, good, say better. Diencephalon, perfect. Third ventricle is at the level of the diencephalon. So tanycytes are at the level of the diencephalon. So all the thalamuses, right? So thalamus, epi, sub, whatever. Okay, impulse conduction. This is homework for you. You have to make sure that you remember how action potential in a neuron is propagated. So what is the sequence? What do you do, etc. Quickly, just I want to get your attention here. You know that it's like a graph that you draw, right? And it's like, I remember how is it? Something like this. Okay, so that's like an action potential, because in the heart, it's like this. Okay, so in the neuron, it's a bit different. Could you tell me the most positive voltage and the most negative voltage of a plasma membrane in a neuron?
Plus 30 and minus 70.
Perfect. Okay, it's going to be plus 30 right here, millivolt, and minus 70. Okay, so minus 70. Good. Okay, origin of the neuroglial cells. Microglia we have talked about, so bone marrow monocyte precursor, because it is a phagocytotic cell. Everything in relation to PNS, neural core cells. All the neuroglial cells, neural tube, and all the CNS, neural tube. Okay, so both of those things will come from a neural tube. Let's talk about PNS. Okay, so a bunch of things are related to the PNS. So we have a bunch of nerves, right, ganglions, etc. So let's kind of have like a quick overview of what is happening, specifically for the peripheral nervous system. So here you have a spinal nerve, right? Here you have two roots. We have the ventral root and we have the dorsal root. And this is your spinal cord. So in spinal cord, it's the opposite. The outer is the white matter, the inner is the gray matter. And this area is known as the anterior horn of the spinal cord. And this area is known as the posterior horn of the spinal cord. Within the dorsal root, you have a structure called dorsal root ganglion. So can we quickly do a thing with the afferent efferent situation? So afferent nerve is another name for it.
Sensory.
Perfect. You're going to see that sensory branch is going to be coming into the spinal cord. It comes in, right? And what's going to be happening here in the spinal cord? What should this sensory branch do?
Doctor, I have a question.
Yeah.
Can you also say that there is autonomic?
Hold on. So we're simplifying and now we're going to add. Okay, so you're going to see that sensory branch, sensory information, sensory nerve is coming in from the periphery all the way to the spinal cord, to the posterior horn, right? To the dorsal posterior horn of the spinal cord. What should be happening here?
Synapse.
Synapse. Synapse with what?
Postsynaptic cells.
Interneuron. It should synapse with the interneuron. Interneuron will take you places. This interneuron can fully avoid the brain and simply connect this sensory nerve to the motor division of the same nerve, which is coming out via the ventral root, right? Or this interneuron could ascend all the way to the brain and synapse with the particular areas in the brain as well that are needed for the perception that I'm touching something, right? So most important things is that in the posterior horn of the spinal cord, there is a synapse happening. A synapse between what and what? A synapse between the sensory, okay, sensory and interneuron. Okay, so sensory neuron and interneuron. Sensory neuron is a which kind of neuron? What is its type?
Pseudo.
Pseudo-unipolar. How does pseudo-unipolar neuron looks like?
One axon that will divide into two.
Well, then it is. Okay, never mind. Okay. Okay, so yes, so it's going to be something like this. Okay, so this is the cell body and this is the axon of the pseudo-unipolar neuron. Where is the cell body of the sensory neuron? Where is it? Dorsal root ganglion. Very good. So you can see that there is this structure, this thickening in the root. It's called dorsal root ganglion. What do we have here? Cell body of the sensory neuron. Where is its axon?
On both sides. One to the spinal.
Very good, very good. On both sides, very good. It's on both sides. It is where the spinal nerve is, and it is where the dorsal root as well. It is a one long axon, right? So you can see it's on both sides. So a question, is there any synapse happening in the dorsal root ganglion?
No.
Absolutely not. Dorsal root ganglion possesses 0 synapses. There's nothing to synapse with, because where does the synapse happen?
In
the gray matter.
But specifically, how do we call this?
Posterior horn.
Posterior horn of the spinal cord. Okay, so the synapse of the sensory neuron will be happening in the posterior horn of the spinal cord. Is it clear?
yes.
Okay, one more thing. Where are the dendrites? On which side? On the left or on the right?
On the posterior horn.
Where are the dendrites? On the left or on the right?
On the right.
On
Right.
the
The
right.
cell of the right.
They're on the right because they're receiving things. Yeah. So dendrites will be here. somewhere where you're touching things, I don't know, smelling, tasting, whatever, sensory. Okay, so dendrites will be here, of the pseudo-unipolar neuron will be here. Where is the axonal terminal? It's on the left, it's synapsin, it's here, right? So it is within the posterior horn. So that's where the axonal terminal will be. Is it clear?
Yes.
Okay. So that's what we talked right now, that we have a sensory neuron. Sensory neuron could be of two types. It could be somatic or it could be visceral, afferent. It doesn't matter. Okay, you remember somatic is from the limbs, visceral is from the body, within the body, right? So it doesn't matter which division, it comes in like that, somatic or visceral. They're going to be coming in like that. Now, we have here interneurons. As you can see, they're kind of connecting my sensory neuron to my motor neuron. And you can see that the motor neuron are of two types. We have the somatic motor neuron and we have the visceral, so the autonomic one. And what's most importantly is that only the somatic motor neuron, only the somatic efferent neuron is capable of synapsing with interneuron. So this is the reflex arch that we talk about, like patellar reflex or something like this, where the sensory neuron synapses with interneuron, which in turn synapses with the somatic efferent neuron. Is it clear?
Doctor, so the autonomic have to go to the spinal cord or the brain.
Autonomic is autonomic. Your interneurons have nothing to do with it. Autonomic is part of your sympathetic, parasympathetic situation, right? So you're going to see that when we do spinal cord, do you see there is additional horn here?
Yes.
It's called lateral horn of the spinal cord. And that's where the sympathetic part of the autonomic innervation will be. So that's where autonomic or visceral efferent neuron will originate from. So there is no interneuron in this area. So this lateral horn is the sympathetic part of the autonomic nervous system, but we will talk more details next class.
Yes.
So again, sensory neuron can synapse via interneuron only with somatic efferent. It cannot synapse with autonomic. Autonomic has its own area in the spinal cord, which you call lateral horn. Does it make sense?
Yes.
Oof, okay, so let's talk about the what did I want to talk about? Yes, this part. Okay, so the cell bodies of the motor neurons. So I want to get your attention because that's the question that they'll typically ask you. Where the cell bodies of the sensory neurons are? Where the cell bodies of the sensory neurons?
Dorsal root ganglion.
So it's going to be dorsal root ganglion. Okay, so that's where the cell body will be. Cell bodies of the sensory neurons are sitting in the dorsal root ganglion. Where are the cell bodies of somatic efferent neurons?
On the spinal cord or the CNS.
Basically CNS, brain, brainstem, or spinal cord. So the cell bodies of the somatic efferent, they're sitting in the CNS itself. But I repeat, dorsal root ganglion is PNS. It's not central nervous system, it is peripheral nervous system. So that's the major differentiation that they want. Is it clear?
Yes.
Okay. And lastly, we have the autonomic, the visceral efferent. Where are their cell bodies?
It is
They're in the organs.
Say better. The correct answer would be depends sympathetic or parasympathetic. Because sympathetic, right, will be both in the lateral horn and in certain ganglia. We call them pre- and paravertebral ganglia. That's where the sympathetic division would be. Parasympathetic ganglia are near the organs. Yes. Parasympathetic
sacral?
are near the organs.
Doctor, is it cervicosacral?
Again?
The parasympathetic, is it cervicosacral?
No, cervical sector has nothing to do with neither. We don't have sympathetic or parasympathetic in cervical. We'll talk about it next class, okay? But it has
Yes.
nothing to do. You're confusing.
Yes.
Mhm. So is it clear for now, guys? So sympathetic of autonomic motor neurons are where?
Depends.
But it doesn't, because I'm asking about sympathetic. So sympathetic cell bodies of autonomic motor neurons are where?
Prevertebral and paravertebral.
Ganglia and
Lateral
horn. Two places, these ganglia and lateral horn. But parasympathetic are in the ganglions near
The organs.
organs. Is it clear? Okay, so this is what we have talked about. Just read some additional things, right? So sensory ganglia are going to be a bit outside. Okay, speaking of nerves, so I guess you understand that spinal nerve is a very interesting structure because technically it contains two divisions, right? So it has the sensory component and it has the motor, the one which is living, spinal nerve itself. And then the peripheral nerve will of course be divided in a way. So peripheral nerve, this is what you can see here. It's a cross section of it. Its composition is actually very similar to muscle. So basically what you see is here, these dots are axons. Each of this dot is an axon. Outside of this axon, you can see that there is endoneurium. So this is a loose connective tissue which surrounds individual nerve fiber or axon. A bunch of axons are put together into a fascicle, right? Like a nerve fascicle. This nerve fascicle is surrounded by the perineurium. Perineurium is specialized connective tissue. Another way, you could also say reticular connective tissue. So also loose, also reticular, also specialized. There'll be just one difference. We'll talk about it right now. A bunch of fascicles are put together and surrounded by this tissue. This tissue is called epineurium. This is a dense, irregular tissue, which kind of puts all of the nerve fascicles together. Epineurium is the one that possesses blood vessels and adipocytes, surprisingly. Okay, so you will see that fat is also present on the nerve. So this is what we have talked about, right? So these are the individual axons. You can see that they're surrounded by the endoneurium, just loose connective tissue, nothing interesting. Fibroblasts are sparse. So this loose connective tissue was secreted by Schwann cell itself. Endoneurium was created by the Schwann cell itself. It's a loose connective tissue, which was created by a Schwann cell. This bunch of axons are being put together into a fascicle, right? And you can see that there's some kind of tissue outside of the fascicle. You call this perineurium. Perineurium has a very particular cells. You call them perineural cells. Perineural cells are a combination of epithelial and smooth muscle cells. So in other way, they are myoepithelial cells. Perineural cells are myoepithelial cells. They're sitting within here, right? So this is going to be the perineurium, which is kind of outside of a nerve fascicle. Because these cells are present there, perineurium acts as an active diffusion barrier. So it makes sure that nothing can get to the axon, so nothing can be toxic for them, right?
I have a question, doctor.
Yes.
Is it by preventing the plasma blood to be mixed with the into neuron?
Not really, no. So like whenever we say that this is a blood nerve barrier, we don't necessarily mean that blood itself should not go here. We mean that whatever is in the blood, like toxins, bacteria, viruses, they should not be able to cross this layer and get here, but not the blood itself, yeah? So if the blood surrounds this nerve, it's fine. But if this blood contains something pathogenic, then it's not fine. So we're creating this barrier.
Yes.
Perineurium is the barrier. Okay, and lastly, the epineurium, that's just the dense, irregular connective tissue. I told you a bunch of blood vessels, a bunch of fat, and etc. Okay, so let's take a look at our first slide. Yay. Okay, so peripheral nerve, H and E. So cross section. How do you call this?
Vesicular.
Fascicles. Okay, so these are going to be the nerve fascicles. What is inside of these nerve fascicles?
Axons.
So each of these dots is going to be an axon, a nerve fiber. What surrounds the axon?
Endoneuron.
Very good. Endoneurium. Okay, so this tissue that surrounds the fascicle is perineurium. Okay, so this is going to be the perineurium. And everything where you see the blood vessel adipocytes, this is epineurium. Okay, it's going to be epineurium. So I point, you tell me. So this.
Epi.
Epi. Okay, this is epi because you can see the blood vessel and you can see adipocyte. Do you see surrounding the fascicle, there is like a red line?
Yes.
Mm-hmm.
It is the peri.
Peri. Okay. Yes, peri. This is very clearly visible here as well. What is this?
This is the epi.
Epi. But the dark red is?
Peri.
Peri. Could you tell me why peri is darker?
Because of the nucleus of the pericytes.
Nope. If you magnify, you see that.
Because of the collagen?
Where is
specialized collagen.
Nope.
Is it because of the external lamina?
Nope.
Schwann cells?
Nope. So this is, again, what is this?
Epi.
Epi. Okay, so this is going to be epi. What about this?
Epi.
Peri. Okay, so this is going to be peri. So which one is darker, epi or peri?
Which one is which?
Peri.
This one is epi. Okay, so this one is outside. This is epi. This one is peri. So P.
Yes.
Which one is darker?
The peri.
Peri is darker. My question is why?
Because peri doesn't have fat.
But fat is white.
Is it because it is
okay,
red?
I see your point, Mika. Okay, so no.
Because it's dense. Dense connective tissue.
So if it is denser, it should be darker, but you see that it is lighter.
Oh.
So epi is lighter than peri. Okay, you guys don't know. This, which cells are these?
Pericytes?
Nope. They're called perineural cells. And perineural cells are which kind of cells? Myoepithelial cells. They possess lots of actin and myosin, which are proteins. They're filled with proteins, so they're much darker, much redder, right? So the answer is because of the perineural cells. Okay, what are these?
Axons.
What's this?
The epi.
This is the epineurium, dense irregular connective tissue with a bunch of adipocyte blood vessels, right?
Yes.
The fibroblasts are not shown here in this section.
Again?
The fibroblasts, like you said, there are spurred fibroblasts in the section, so it's not shown in this section.
Sparse Fibroblasts are in the endoneurium. Endoneurium is here. You cannot see it, but in the dense irregular connective tissue, you can see fibroblasts. So these cells, all of them are going to be fibroblasts, these ones. Okay, like these ones. These are fibroblasts. In the epi, they are present because this is a dense irregular connective tissue.
Okay.
Want to ask you a question? Let me find it. Okay. Do you see this?
Yes.
So
Yeah.
this is a perineural septa. Perineurium is capable of sending septa down the fascicle. So you can clearly see a very thick, nice structure. Do you see these?
Yes.
Agree with me, they're much thinner than these ones.
Yes.
So
Yeah.
those are perineural septule. So
Yes.
these are the septule. Professor Wolf was talking a lot about them last year. So I feel like it's important. So septule is much smaller than the septa, right? So it's much thinner, better said.
So the fascicles are they are lobulated because of the septa, or?
No, okay. It's very good that you're thinking about that. Gland is lobulated, but fascicles aren't. It just happens. Like you just see occasional septule and that's it. But no, nervous tissue cannot be lobulated. Gland can be. So there it would be perfect. If it was an epithelial cell sitting there, would have been perfect that yes, that tissue is lobulated. But this is not the case. Okay, this is a nervous tissue. It can't be lobulated. Even though I see septa and septule, I cannot say it's lobulated. Okay, so that's the septule that we have talked about. Okay, so this is like beginning of a septa because you can see it's nicely going down. But these little thin things are septules. Okay.
Yes.
So let's magnify a little bit. What are these?
The endo.
So hold on. So let me highlight what I want from you. So these darker things inside.
Oh, microtubule.
Oh, you need an electron microscope to see that. This is a light microscope. You will not be able to see it. This is an axon itself. So the darker part, that's the axon. But I think I told you, we don't say axon, we say axoplasm. In our university, you will see that they'll point here and they will say this is the axoplasm. What is the white tissue outside?
In the endo.
Myelin sheath.
Myelin sheath, very good. Okay, so don't forget, right on the surface of the axon, there should be myelin sheath. So this white space that you see outside, this has to be the myelin sheath. Now, Muhammad really wants to say endoneurium, but this is H and E. Can endoneurium be seen with H and E?
Doctor, but I have a question.
Just a second. We'll first answer this and then we'll get to your question. So can endoneurium be seen with H and E? Endoneurium, which tissue is it?
Which tissue? Loose.
Connective.
Loose connective tissue. Which fiber is dominant in loose connective tissue?
Reticular fiber.
Reticular fiber. Can reticular fiber be seen with H and E?
No.
Not really.
No. So let's take a look. This is H and E, right? Last time we checked it was H and E. So can I see endoneurium? I cannot. Okay, you cannot see endoneurium. So this on the surface is not an endoneurium. Professor Wolf, he says that this is a plasma membrane of a Schwann cell. Okay, so this is a plasma membrane of a Schwann cell, but not endoneurium because endoneurium contains reticular fibers. So to see endoneurium, you will need heavy metal, right? You will need heavy metal to do that. One more thing, and then we'll answer your question. These big nuclei that you see, do you see? So this is like an axon, and on the surface, this is a nucleus, right? Axon, then nucleus. Whose nuclei are these?
Satellite.
But satellite is around cell body. This is an axon. and then
Schwann
on the
cell.
top of it,
Schwann cell.
of course, it has to be the Schwann cell. Okay, so these nuclei that you can see, these are going to be the nuclei of the Schwann cells. So this one, this one, this one. Okay, so this one, they're nuclei of the Schwann cells. Okay, go for your question.
Yes, actually, I wanted to ask you, how can you see the myelin if the lipids will be removed by HU? But I know that it's already removed.
Exactly. It is already removed. This white space, absolutely correct. Technically, it's not myelin. It's a white space. The lipid is gone there. Okay, it's not there, but you just say that this is a myelin sheath, but absolutely correct. In reality, it's not there. It's just a white space. It was fully lost.
Yes.
Yes. Okay, so I point, you tell me, what are these?
Axons.
Exoplasm.
Nope.
Schwann cells.
Say better.
The
Nuclei of the Schwann
Nuclei
cells.
of the Schwann cells, yes. Okay, so these are the Schwann cell nuclei. What is this?
Exoblast.
Exoplasm, the white space,
Myelin sheath.
myelin sheath, right? So brackets, myelin sheath, and outside is a bit of a plasma membrane. Okay, what is this?
Septule.
Septule, very good. It is the perineural septule. Which cells are these?
Type 3. No,
Perineural.
Perineural cells, okay, and they are which kind of cells?
Myoepithelial.
Myopithelial cells, perfect. They are myopithelial cells. Type 3, Mohammed, that's the
Acidic.
exactly, that's the GFAP, the glial fibrillary acidic protein. Okay, osmium. What is osmium?
Heavy
Heavy
metal.
metal.
Heavy metal. The full staining of this slide, this is also a peripheral nerve. It is osmium tetraoxide plus hematoxylin. So what is the purpose of the slide? To see anything that has lipids, right?
Yes.
You should remember that osmium tetraoxide is colorless. It has no color. Yet you see there's a lot of dark color. So how do we see it? Because it becomes oxidized, right? So it has to become oxidized to dioxide, and that will create the black color. So what is the white thing?
Myelin sheath.
Nope.
The axon.
The axon itself or the exoplasm?
Exoplasm.
The brown thing?
Myelin.
Myelin, yes. The brown thing is the myelin. Now, do you see the dark line?
Yes, it is into. Yes.
Neurium. Endoneurium. Okay, so the dark line is the endoneurium. I know you might have questions because, like, where is the nucleus, right? So that's the nucleus of the Schwann cell, and then we see that it still continues with it. Like endoneurium continues with it. I cannot tell you much, guys, honestly. Like,
Doctor, what is the black point there? Little bit in the left up.
to the left up, these ones?
Yes, yes.
Nothing, it's just an artifact, okay? So this could be like the Schwann cells themselves, okay, that are like the rest of the plasma membrane there. It's just a bit of an artifact, just extra areas where the dye precipitates, but it's something very lipidy, okay? So Schwann cells are super lipidy, so it could be like a part of a Schwann cell. According to Professor Wolf, the white space that you see between the axons, this is actually a cytoplasm of the Schwann cell. Okay, so that's what he says. This is the cytoplasm of the Schwann cell. So maybe that's like the rest of the Schwann cell. But the most weird thing is that this is endoneurium. So that's the accepted version. So this dark line on the surface, this is endoneurium. We see it there because it
Reticular fibers are stained.
possesses reticular fibers. Yes. Okay, so because it possesses reticular fibers.
Doctor, can I ask a question that confuse me? Yes, it's about the Schwann cells. Like, for example, one axon with one Schwann cells, or a lot of axons inside the Schwann cells? Because
no, it's one axon and a bunch of Schwann cells on the top.
Okay.
Yeah. So if it is myelinated, if the axon is myelinated, if we see this myelin, if we don't see the myelin, it could be a bunch of axons and just one Schwann on the top. So it cannot finish the myelination process.
Thanks.
Okay, spinal ganglion, aka dorsal root ganglion, aka sensory ganglion. 3 names for the same thing. For which thing? This thing. What is in it?
Cell body of the sensory.
We kind of talked about it, right? And these cell bodies are belonging to the pseudo-unipolar, right? So the cell bodies are here. The axon is kind of everywhere. It is part of the PNS, so neural crest cells. It's very weird that we call the cell bodies of the dorsal root ganglion. Yeah, we call them ganglionic cells. We call them ganglion cell. It's very weird because it's not a cell, it's a part of a neuron. It's not a separate cell, yeah? It's just a part of a neuron, yet we call it ganglionic cell. The rest of the things we talked, yeah? So it's everything that we have talked before. So this is why let's take a look. So this is a spinal ganglion, dorsal root ganglion, sensory ganglion, H and E. Outside of it, this is dura mater. Okay, so this is dura mater right here. Don't ask. Okay, so apparently dura mater goes all the way here. Apparently it's not just on the surface. Apparently it goes all the way here. So do not ask. I say it, but deep down everything is like, you know, I don't want to say it, but that's what they want. This is dura mater. Okay, this is nerve of some sort. I don't know. It could be a dorsal root nerve or it could be a spinal nerve. We don't know which way are we going. Outside of this nerve, we see epineurium. So this part right here, this is epineurium. So you can clearly see how epineurium is continuous with the dura mater. This nerve is a bunch of axons, right? So these things that you can see here, these are axons. Inside of the spinal ganglion, you can see these circles. These are the cell bodies of the pseudo-unipolar sensory neuron, right? So here they are. We call them ganglion cells or ganglionic cells. I repeat, it's very weird because it's not a separate cell, it's just a part of a neuron. So what is this?
Nucleus.
Nucleus. Okay, so nucleus. What is the dark thing?
Nucleolus.
Nucleolus. This is the cytoplasm. You can see a bunch of blue dots.
Nissl bodies.
Nissl bodies, very nicely seen here. Okay, so these are the Nissl bodies. Some kind of cells on the surface.
The satellite.
Satellite cells, yes. Okay, so these cells on the surface, they are satellite cells. Dura mater. How from this picture alone, from this picture alone, you can say that this is actually a sensory ganglion. It's not sympathetic or parasympathetic or anything like that. How do you know this is a sensory ganglion?
From the satellite?
No.
Does it have to do something with the axons? There's only one or something like that.
From the cell body?
From the dura?
Something with the axons was very close. Okay, so there is something that in the sympathetic and parasympathetic ganglion, there is something happening there that is not happening here.
The synapsing.
Synapsing. And what do you need for synapse?
Dendrites.
Exactly. Can you see dendrites?
No.
No. So there is no synapse. Okay, so there is no, because on this picture, these are axons. I can see axons. I can see cell bodies. The one thing I cannot see are dendrites, and I need them for synapse. So just from these two pictures alone, you can easily say that this is a sensory ganglion because there is no synapse happening there. Where is the synapse of the sensory neurons? Where is the synapse?
In the dorsal ganglia?
But this is dorsal root ganglion, and we just said that there is no synapse.
Somatoaxonal?
Nope.
In the horn, the
I
posterior
agree.
horn.
In the posterior horn, perfect. In the posterior horn of the spinal cord, that's where the synapse is. What was the sensory neuron synapsing there with?
Interneuron.
Interneurons, perfect. This ganglion is non-synaptic. There's no synapse here whatsoever because you can't see the dendrites. Okay, speaking of sympathetic ganglion, what are the major differences? We can see them here. So cell body is going to be, sorry, the neuron is going to be multipolar, not pseudo-unipolar. Sensory has no synapses. The sympathetic, parasympathetic has synapses. Sympathetic are in the pre/paravertebral ganglion / lateral horn. The parasympathetic are next to the organs. So we have sympathetic ganglion, and if I'm being correct, this is the paravertebral ganglion. So last semester, you should be able to see them, right? So right next to the vertebrae, you can see a bunch of paravertebral ganglions there. The method of preparation here is Bielschowski. Bielschowski impregnation is a simple, like a fancy way of saying silver impregnation. So in neuro, silver is Bielschowski. So you'll always see it. So what are these? Cell bodies of motor neurons. What are these?
Nucleus.
Nucleoli. Okay, so these little tiny things there, these are the nucleoli. This is the nucleus, sorry, but the dot is the nucleoli. Do you see the dot? I think I covered it. So here's the dot, yeah? So nucleoli are visible there. Okay, now comes the fucked up, I apologize. Now comes the bad thing. Okay, so the bad thing is that we have to identify where's the axon, where's the dendrite. This slide is not for that, yeah? We shouldn't, like, you won't be asked, but who knows? Maybe this is the year that they do. Okay, so let's try to identify. What are we looking at? So what do you think this thing is? Yes, I agree with you. This is an axon. Why is this an axon?
No branches and very long.
Very long, no branches. And lastly?
Myelinated.
But how can you see if it is myelinated?
But it is branching, Yulia, it's branching, right?
This is not a branch. This is not a branch. I can't magnify it more, but when you're studying, you'll see this is not a branch. At the end, this is not a branch. It's very straight, and everything is what looks like it is branching, in reality is behind it. Okay, so you can see there's a faint color here, yeah? So it's not an actual branch. It's like an area. It's like an area which is behind the thingy, okay? It's like a behind the axon kind of part.
Doctor, it must be straight.
Yes, I agree with you. It must be straight, but sometimes we aren't lucky to see like the whole straight thing, you know? Yes. The last thing that I want from you is the consistency, okay? So the thickness consistency, that it is thick throughout, okay? So it's going to be thick throughout. Like this thing, is it an axon or a dendrite?
Dendrite.
What makes you think so?
Consistency is not available.
Because you think that it's thinning out, right? It looks like it is becoming a bit thinner. Yeah, I agree. It's very difficult to find.
Doctor, the middle, I mean that red circle have a straight line up.
This one?
Yes, go up, there is a straight line.
This one?
Yes.
Yes, okay, I could think, but I don't like this part. It looks like it's coming out from it. Okay, so it looks like it is actually coming out from this part. So maybe it could be a dendrite. But like, let me try, like, which one do you have more? Axons or dendrites?
Dendrites.
So majority of them are dendrites. All of these lines that you see here are dendrites, but it's very difficult to find like a good axon, right? So here, for example, do you see? It's sort of thick and sort of straight and uniform, right? So this area, so it could be an axon. This area, this is definitely a dendrite. It started to become thinner, right? So it started thick and now you can see that it is thinning out, right?
Doctor, what about the most left and down red circle? It has a right, the left corner. I mean down.
This one.
There is a straight line.
But we talked about this one.
Ah, sorry.
You're the one who said that this is the axle. Okay, a bit more. These ones.
Is it dendroid?
Yeah. Very branchy, do you see? Very nicely, you can see they're branchy. This one.
That's
Sorry,
an
I drew
axon.
on it.
Axon?
An
I
axon.
wouldn't, because it's a bit thicker. Okay, so the area where it starts, it's a bit thicker, and I can't erase it. I apologize. Okay, these things that are beginning here.
It is so short and.
Yeah, but you can see how thick they start, and they're already becoming thin.
Yes.
Dendrite. They're all going to be dendrites, yeah?
Yes.
This.
It's a dendrite.
Dendrite.
Doctor, there is one in the very right. Yes, there is one. Yes, here there is a straight line going down.
This one?
No, the above it. I mean more right to it.
This one?
No, more right.
This one?
Like this. Yes. No.
Okay. I don't see it. Sorry. So these ones, what do you guys think? All of them here?
Dendrites.
Yeah, for sure. All of them here are dendrites. From here, somewhere here, you can see that there's also a bit of a branching. Okay, so like this. So it also has to be dendrite. So axon probably is on the other side that we cannot see. But as I have said, yeah, so this slide is not for axon identification. Oh, wait, I found a very good axon. Okay, so here. Okay, so it's a good one. So it's more or less straight. There isn't much branching going on. Okay, so this is a good axon. In cerebellum, when we do cerebellum, that's where we're going to be looking for like long axons there. Okay, guys, that's it for today. I'm missing 15 minutes. I will add them to the next class. Yeah.
Thanks a lot, Doctor.
I just have a question. The last question for the last slide. How could this dye like show the axons and dendrites? Like, what is this dye exactly?
This is silver. Silver shows lipids. Plasma membrane of a neuron has lipids. So dendrites, they are covered by a plasma membrane. So they must have lipids, right? Axon as well, it has lipids, so you can actually see it. So silver shows lipidy things.
Okay, I got it. But it's not another name. It's another name for silver impregnation. Like it's still silver, but it's not another name.
Exactly. It's the same silver, but you're gonna call it Bielszowski.
Okay.
Okay.
Perfect.
So yeah. So part 1 is done. So see you next.
Doctor, can I ask a last question if you